Good morning, students. We shall start with the next chapter in ethology. In this session, we'll be doing about pheromones. What do you mean by pheromones? What are the different types of pheromones? And you can see examples in insects and vertebrates which secrete pheromones. What are pheromones? In the year 1975, Novak described them as chemical messengers. These pheromones are chemical substances which are released into the external environment. It is released by an organism which is called as a releaser, and it brings about a change, or the message which is released along with the pheromone is picked up by another member of the same species. So it is referred to as intraspecific or species specific. One organism is releasing a pheromone, which is a chemical substance which carries a message, and this will affect another organism. When you say it affects another organism, it brings about change either in the behavior of the second one, or in the process of development, or in the process of reproduction. So one is releasing the chemical substance pheromone which brings about change in the other organism in its behavior or development or reproduction. So when we say the pheromone is giving out some message, what message is that chemical giving or passing on to another member? It provides information like the sex of the organism which is releasing the pheromone, whether it's a male or a female, or it can also give information about the reproductive status of the individual, whether it is reproductively mature or not. Now all of you know about hormones. Hormones are also called as chemical messengers, but hormones are released inside the body and it brings about some change within the same organism. So the hormone released in one part of the body is carried to another part which is called as the target organ, and this organ will bring about some physiological changes like release of a enzyme or release of a digestive juice or anything. It brings about change in the physiological responses of the same organism. So hormones are chemical messengers which are bringing about change in the organism in which it is released, whereas pheromones are released into the external environment and it brings about change in another organism. So the pheromones are released by exocrine glands. They have duct, so along with the duct the secretions will be released. In the year 1959, Carlson and Lucia described about the pheromones. The term pheromone is derived from two Greek words, pherein meaning carry and harmon meaning to excite. So pheromones are hormone-like substances secreted by exocrine glands released into the external environment. So it is called as ectohormones. As it is released outside, it's called as ectohormones. They will act like chemical messengers. They carry signals, and these signals in turn brings about responses in the members of the same species. So you call this response as intraspecific communication, wherein the release is trying to communicate some information with the receiver. So the one which is releasing the pheromone is the releaser, and the one in which it brings about change is the receiver. So there is a communication between the releaser and the receiver, which we call it as intraspecific communication. There are different types of chemicals which act as signals. So let us say there is a chemical which is bringing about a communication between two different species. So earlier we spoke about the chemicals which is bringing about change within the same species, that is intraspecific or conspecific. Here we are talking about chemicals which are bringing about changes or responses in two different species, then it is called as interspecific. So there are certain chemicals which are capable of bringing about changes between two different species. So the first type of such a chemical is called as allomone. In this type, there is an organism which is emitting. So the emitter which is releasing the pheromone is benefited. So the pheromone is favoring the emitter. So let us say there is an organism. It has got threat of being eaten by the insect. It releases the allomone. This allomone will act as a repellent of the insect. So it is protected by being eaten up by the insect. So it is between two different species and it is called as allomone. The second type of chemical is called as Kairomone. Here it favors the receiver. An organism is secreting the pheromone which is received by another one, and the receiver will actually get benefited. For example, if you look at a parasite cannot survive on its own. It needs a host. It is always in search of host. Now this parasite can recognize some chemicals secreted by the host which will attract the parasite towards the host. So the host is secreting chemicals for some other reason, whereas the parasite is getting benefited by identifying that chemical, and it gets attracted and it gets attached to the host. Such chemicals are called as Kairomones. So now there are different types of pheromones. Basically there are three types. The first type is called as releaser or signaling pheromone. So this pheromone is released by one organism. It follows a pathway which involves central nervous system, or you can say it as neuro-humoral pathway. So the pheromone is picked up by the sensory cells. It is taken to the central nervous system. Then it reaches the blood. It follows the neuro-humoral pathway, and an organism will release a pheromone which will act as a signaling chemical, and this signaling chemical will be perceived by another organism, and the other one will know information about the first one, which is the releaser. The first one is releasing the pheromone which is carrying some signal which is perceived by the receiver. So the first one, when it is releasing the pheromone, it gives information about itself, like the sex of the individual, whether it's male or female, the species, the sexual status, whether it is reproductively active or whether it is taking care of the young one. So this type of pheromone is called as releaser pheromone or signaling pheromone, which is released by a releaser, picked up by a receiver. It is picked up by the sensory cells, then it follows neurohumoral pathway and brings about responses in the receiver. Aggressive behavior which is seen in female. Suppose a female has given birth to young ones and a male approaches this female, or a different male which is not its mate approaches this female, she shows aggressive behavior. She releases a pheromone called a sex pheromone which will try to chase away the male which is not familiar to the female, which tries to come near this female or encounter this female. This is called as aggressive behavior and it releases the pheromone, sex pheromone. Another classical example what you see in the animal world is the alarm pheromones which are secreted by some insects. Alarm pheromones are those chemicals which are responsible to raise an alarm when there is threat to the colony. It is seen especially in those insects which have got social life where they live in colonies. To name a few colonial social insects, we have ants, honeybees and wasps. All of you know that if you try to disturb a honeybee colony, then one of them will come and sting you, and when they sting, they release alarm pheromone. This alarm pheromone will be picked up by other members of the colony and they all try to attack you. How did they get the information that there is threat to the colony? When the first one bites you, it is releasing alarm pheromone which attracts other members of the colony, and their main goal is to protect the colony and chase the predator away. So this is again another example for releasing or signaling pheromone which is exhibited by the colonial social insects. The second type of pheromone is called as primer pheromone. Unlike the first one, the first one releaser pheromone, the second one is the primer pheromone. This one will bring about long-term effect in the receiver. It acts on the endocrine glands and brings about physiological responses in the body. Let us see an example. If you look at the rat reproductive cycle, they show estrous cycle, like what is there in the human species menstrual cycle. The estrous cycle in the rats, suppose there is a cage in which only female cycles are there, and these females will be releasing certain pheromones. Because of the effect of the pheromones, the estrous cycles become irregular. Now, if you introduce a male releases some pheromones, and these pheromones will act on the reproductive organs of the female, and it regularizes the reproductive cycle of the female and it normalizes the cycle. So the female reproductive cycle becomes normal and it is ready to mate the male. So in this case, the pheromones released by the male is acting on the reproductive glands of the female, and it is regularizing the estrous cycle of the female so that they are ready to mate. So the pheromone is bringing about a long-term change on the endocrine glands, bringing about physiological responses. So this is one example for primer pheromone. There was another experiment conducted by a scientist, Bruce, which is called as Bruce effect. In this experiment, when a female rat which is pregnant, which is being kept in a cage, now you remove the male which was responsible for the pregnancy of this female, you remove that male and introduce a new male into the cage. When a new male was introduced into the cage, then what happened to this female? She induced abortion and she aborted the developing pups. After abortion, she was ready to mate the new male. This was studied by Bruce and is called as Bruce effect. Why did the female do so? The female induced abortion of her own pups because if she had waited for the completion of the gestation period and if she had reproduced, then the new male who did not father these pups would have eaten off the pups so that he can mate the female and produce offsprings of his own. So the female knew this behavior of the male and hence she induced abortion. Anyway, these pups will be killed by the new male. So she induced abortion and she became ready to mate with the new male so that the progeny will survive. This is another example for primer pheromone. If you look at honeybee leads a colonial life. In the colony, there is a single reproductively active female called as the queen. This queen, when she is there, she will not allow other females to become the queen. She suppresses the development of reproductive organs of other females by producing queen substance. This is a pheromone which is released by the queen bee called as queen substance, which suppresses the development of the reproductive organs of other female members of the colony. Similarly, there is also social pheromone released by the termites. So these things we will be doing in detail when we do social life in the next chapter. The third type of pheromones is called as imprinting pheromones. So this is very important, like what we studied in the previous chapter. Imprinting is a critical state where the mother will bind with the offsprings when they are born, because the offsprings need to be taken care of. They have to be fed and protected until they become independent. So for this to happen, they have to bind with the mother, which is called as the filial imprinting. And if you look at mice and rats, when the young ones are given birth to, the young ones are in a very immature state. They will not have the development of fur, the skin will be naked, the eyes will be closed. And how these immature pups will recognize the mother is by the identification of the pheromone secreted by the mother. Mother releases the pheromone, and when the pups will lick on the secretions of the mother, they bind with her. These pheromones are called as imprinting pheromones. So these are the three types of pheromones what you come across in different animals. Apart from this, the pheromones are also classified as volatile and non-volatile pheromones. This is based on the molecular weight of the chemical substance. If the molecular weight is low, then you call it as volatile pheromone, and this is responsible for short time effect, like signaling. When there is a danger, it has to produce an alarm, which is a short time. It is required for a short time. So this is a short time effect pheromone. Or suppose some insect has to attract the opposite sex, then it will release the pheromone for attracting the opposite sex. So these short-term effect pheromones are having low molecular weight and they are called volatile pheromones. The second type is non-volatile, where you have the chemical substances which have high molecular weight, and this generally has long-lasting effect, which will be seen in the wild animals where they have to mark their territory. Generally, if you look at the animals living in the wild, they select a suitable, convenient place for them which has got food resources and where they can guard their young ones. They identify such a place and they mark their territory with the help of the pheromone. Once they mark the territory, they do not allow other wild animals of its own species or any other species to enter into its home range. So this is done by the pheromones which are having high molecular weight, and you call it as non-volatile pheromones. So with this we are done with the introduction and different types of pheromones. Now we shall see some examples in insects as well as vertebrates. We shall start with insect pheromones. Lot of studies has been done in insects where people have deciphered many insects which are releasing pheromones, and if you look at the variety of glands present in the insect, you will be really fascinated to look at how the different cells are modified to become special glands to release the pheromones. We shall see some examples. If you look at the locust has got some epidermal cells which become specialized to release the pheromones, which acts on the nervous system. So if you look at the insect nervous system, there is a part of the brain called corpora allata. So the pheromone will act on the corpora allata, and what does this do? It brings about maturation of other members of the colony when they are. So these epidermal cells will release the pheromone which acts on the corpora allata, a part of the insect brain. And what does this do? This will bring about maturation of other individuals of the same species. It speeds up the process of reproductive maturity, and that's how the pheromone acts on other members of the same species. In Lepidoptera, where you have butterfly and moths, males have got a gland called a scent gland, and this scent gland releases the pheromone. And if you look at the wings have special glands which are called as androconia, and from this the pheromones will be released. And what does it do? It induces the process of mating. So it brings about mating between the opposite sex. Then we have Danus gillipus. Here the male will secrete the pheromone. It brushes the pheromone onto the body of the female with a small brush-like structure called as hair pencil. So with the help of the hair pencil, it brushes the pheromone on the female and brings about mating. In the Bombyx mori, the silk moth, female will produce a pheromone from the saculi lateralis. It secretes the pheromone bombicule, and the function of this bombicule is to attract the male. Another classical example is seen in the female emperor moth, scientifically called Endia pavonia. Here the female releases the pheromone to attract the male, and the male can sense the pheromone, and if the male is 13 kilometers away, even then it can sense the pheromone and it gets attracted and comes to the female to mate. So it is so strong. Bombicule is the pheromone secreted by Endia pavonia is so strong that it is capable of attracting the male, which is 13 kilometers away. So in this you can look at the different organs which are specialized to release the pheromone. In some insects there is a scaly patch, in some there is androconia, in some there are special glands like the Duffer gland, Mandibular gland. And if you look at the ant, there are different types of glands present in the same individual, each one producing a different type of pheromone having a specific purpose. So you can just go through the diagram and you can read what are the specialized insect body part which are responsible for secreting the pheromone. Hymenoptera, where you have the honeybees, when we do social life, you will see that lot of the social life is controlled by the release of the pheromone. If you look at honeybees, in honeybees, they have mandibular glands. The queen and workers, both of them have mandibular glands which secrete a pheromone. They have studied the chemical nature of this pheromone and they have found out that it is 9-keto-decanoic acid. And what does this do? This will inhibit the ovarian development of the workers so that new queen will not be produced in the colony. The entire colony will have only one reproductively active female, the queen, and when she is there, no other females will mature, and the reproductive maturity of these workers is suppressed by the secretion of a pheromone which is known to be 9-keto-decanoic acid. Queen bee also secretes queen substance, and this queen substance will also suppress the development of the ovary so that new queen will not be formed. Queen bee again will produce another pheromone by the mandibular glands, and chemically it was identified as 9-oxodecenoic acid, and the function of this pheromone is to attract the male, which is called as the drone, and during mating or swarming flight the queens and drones will go out for the purpose of mating. So the queen releases oxodecenoic acid to attract the drone, and then the mating will happen. If you look at ants, all of you must have seen how very well organized an ant colony is. If you look at the movement of the ants, they follow a path on which they are moving to and fro in search of the food. All this will happen because of the release of the pheromones. Ants will produce different types of pheromones for different purposes like mating, or finding the food source, or exploiting the food source, or recruiting the nest mates for the purpose of protection of the colony or to keep away the predators. So these are some of the functions for which the pheromones are released. In the ant, IRIDOMYRMEX HUMILIS, what they do is, when they find the source of food, they come back to the nest, and on the way back they leave a trail of pheromones, and other members of the colony will follow this path and find the food source. So they reach the food source with the help of the pheromone which is left as a trail. In Solenopsis, they have PAVAN'S gland. In Myrmica, they have poison gland. In Lasius, they have rectal gland. So in different ants, different glands are specialized to produce the pheromone, like PAVAN'S gland in Solenopsis, poison gland in Myrmica, rectal gland in Lasius. All of them will secrete a pheromone which is released like a trail to identify the location of the food. Leptothorax, here the workers will recruit specific number of the members. So if you look at how the ants are recruited for different purposes, let us take an example of an ant which is trying to get the food back to the nest. Sometimes you must have seen they have found a food source which is quite big than their own body, but some 2 or 3 ants are enough to roll that food to the nest. So when they find the food source, they will also give the information as to how many members are required to bring the food back. So this is done by the pheromones which are called as recruiting pheromones, which will also give the information about the food source as well as the number of individuals required to get the food source. Next, in Solenopsis, here you see that when a large food source is found, they release mass recruiting pheromone so that a group of ants will go and they try to get the food back. If the food source is quite big, many members of the colony will form a trail. Many members will go one behind the other to collect the food and bring it back to the nest. So now we looked at different examples of the ant pheromones. In this picture you can see in the first one the ants are getting the food back. In the second one also they have found a food source and they are getting the food. In the third one it is showing the aggressive behavior where two different ant species are fighting. It could be to guard their nest colony or to chase away the predator. So here there is another interesting thing, what you see in the ants, which is called as slave raid. What do you mean by slave raid? Here there are two ant species. One is Polyrgus lucidus, the other is Formica arcboldi. Now, this Polyrgus lucidus will go to the Formica arcboldi colony and they will confuse the defenders of the Formica arcboldi. The Formica arcboldi has got a colony, and this colony will be guarded by the soldiers of the colony. So, the Polyrgus will go to the colony, they will confuse the defenders, they enter the colony, they carry away the eggs, young ones, and bring them back to their own colony. Here, once they hatch out and once they mature, they will be treated like slaves and they will have to work for this colony. This is called as slave raid. So for this purpose, they also release pheromone for the purpose of slave raid, and the members of this colony will go to the other one and try to get the eggs and larvae, and they will keep them as slaves. So now in Formica subintegra, they also secrete the recruit pheromone for the purpose of slave raid. They have got a gland called as Duffer's gland. The pheromone secreted by the Duffer's gland will confuse the workers which are trying to protect the colony, and when they are confused, they will carry away the larvae and the pupae. Some ant colonies, they secrete pheromones called as colony odors, and this will help in recognizing its own members so that they all can stay within the colony. In another ant called as weaver ant, here it can produce 30 different types of chemicals, and they will be involved in different types of activities. Then you have another special gland called as Nasnoff gland or Nasnau gland. This is found in the worker honeybee in the abdominal region, and it secretes pheromone to recruit the workers. So this is a picture of the slave raid where they are trying to pick up the members of the colony to its own colony. So here you are seeing aggressive behavior in the ant IRIDOMYRMEX purpuris. You can see in the middle there is an ant which has lifted a leg. So this kind of display where when one leg is lifted up means it is aggressive. She has encountered an ant from a different nest. Because of that, she has become aggressive. She is in a mode of defending the colony. So this picture shows the aggressive behavior, and during this a pheromone is released telling that there is a threat because they found a member of another species which has come near this colony. So here this is another picture where you can see the eggs, pupae and the larvae are carried from one colony. You are seeing the recruited ants which are carrying the young ones. So with this we completed the pheromones and their role in insects, different types of glands, and for what purpose the pheromones are released. So basically either it is to locate the food or to defend the colony or for the purpose of slave raid, for recruiting and to chase away the predator. Now we shall look at the pheromones in vertebrates. If you look at the mammals, one important function what they do is the marking of the territory, which is called as the territorial pheromone, and they will have special glands. And sometimes the pheromones can be released along with the urine or the feces, and when it is released it will mark the core area where the animal is living with the help of the territorial pheromone so that other individuals will not be allowed to enter in its home range or the core area of the home range. If you look at the wild animals which live in the forest, for them identifying their home range when they go out in search of food or water, they have to come back to their place where they will rest. It is a difficult task because in wild it is very difficult to locate a region or locate the path. So what do? When they go out from the feeding and resting place, when they go out from the place where they rest and sleep, in the mornings they go out in the search of food. When they go out, they will mark the path with the help of the pheromones. So a lot of studies were done to understand how this is happening. In hippopotamus, what it does when it goes out for drinking water or for feeding, on the way it deposits the dung. When it is depositing the dung, it is moving the tail, so the dung gets scattered to a larger area and thus mark the pathway. In rhinoceros, the feces is deposited on prominent landmarks, like it could be a small stone or a small hill-like structure. It will deposit dung on that so that from a distance it can locate the dung and it can come back to its resting place. If you look at the male tiger, it releases the pheromone tigeramine, which is a fluid which is milk-like and generally secreted along with the urine, and it is found out that it is chemically phenylamine. So this again is used to mark the territory. Another interesting what you can see in the wild cat is it goes near a tree, then from the posterior side of its body, it turns its excretory organ and jets out urine with force onto the bark of a tree. By doing so, every day regularly it will urinate on the same spot. By doing so, it is marking its territory. In spotted hyena, what they do is, wherever they are resting, that place, they will smear the grass with the pheromones released by the subcaudal scent glands and also the feces which is deposited on the stem of the grass. So with the help of the secretion of the scent gland and the feces, they try to mark their territory. The loris, what it does when it urinates, it will catch the urine and it will sprinkle on the tree and hence marks the territory. Capuchin, it will soak its feet and hand with urine, it will urinate, then it will wet its hand and feet in the urine and smear on the leaves of the tree to mark the territory. In lemurs, it will defecate, it takes the feces and smear it on the branches, like as though it is painting the branches with the feces to mark the territory. In greater bush bay, when it is urinating, it will cup its hand, then the urine is collected and rubbed on the trees to mark the territory. Bears will mark by chewing the bark. It will identify a tree, it will chew the bark. After chewing the bark, every day it will urinate on that spot. By doing so, it is marking its territory. So there are special glands in vertebrates for the purpose of secreting the pheromones. In monotremes or marsupials, they have special anal glands and the secretion of these glands are rubbed on the ground. In brown hyenas, they have got two anal glands, white and brown, the secretions of which will help in marking the territory. Male beaver has got anal gland to mark the territory. Opossum, which is a marsupial again, has got cloacal glands. The bear, dog and rat will secrete pheromones with along with the saliva, secreted from the salivary glands and released with the saliva. Another marsupial will chew the twig, and then where it is chewn, the twig is bitten, there it will salivate to mark the territory. Hedgehog will salivate and scratch. Boar saliva has got steroids, and this will induce mating. So when they mate, they stand, and to induce this posture, they secrete pheromone which is steroidal in nature. The female rat will lick its nipple, and it releases the pheromone which will attract the pups which are blind. There are other glands which are studied in vertebrates, like in roe deer, the stag, that is the horn of the roe deer, will secrete pheromones to mark the territory. It also has got metacarpal, anal and glands on the forehead. So it has got glands near the stag, glands on the metacarpal region, glands on the anus and the forehead, which will help in marking the territory. Black buck and sambar have got special glands on the face region. It will rub the facial glands on the tree to mark the territory. Red brocket or solitary deer has got glands at the base of the antlers, that is the horns. At the base they have got the glands which will release the pheromones to attract the females. In elephants, the bulls, that is the male, will have got glands on the temporal region secreting the pheromone to attract the female elephants. Musk deer has got the glands on the region of the stomach which secretes pheromone to attract the females. Humans also do produce pheromones, but it is seen only during the ovulatory period. So if you look at the menstrual cycle of the female, ovulation will happen at around 14th day in a normal 28-day cycle. So at that period, the female will produce a pheromone to attract the male. Similarly, the secretions of the vagina, there is a chemical substance copalin, and this copalin is secreted in huge quantities at the time of ovulation, so that if there is coitus happening at the time of ovulation, it becomes fruitful so that the fertilization will be successful. In males, when the males sweat in the axillary region, armpits, they release pheromones, and this will attract the females. So these are some examples for human pheromones. Thank you. So with this we have completed the chapter on pheromones. So we did the introduction for pheromones, the different types of pheromones, and some examples of insects and vertebrates. We have studied the different aspects. If you have got any doubts, you can get it clarified. Thank you, students.